speak, you can put it in the chat. So concerning the groups, that should be in your schedule. The A groups are for the journal club and the B groups are for our discussions. So I'll give it another minute. And let's see, 42. So we should be there. Okay, good. All right, well, good morning. I'm Susan Mango. Welcome to the second week of the block course for developmental biology. We had something completely different planned, and we reorganized it and changed everything for the lockdown situation, and we think you're going to like it. This will focus on the nucleus, chromosome organization, and learning how to trace chromosomes in cells. As I said a minute ago, if you have a question, for now you can write in the chat area, but once I go into the share screen mode and show you slides, I can't see that, I think. Or if anyone knows of a way to see both, let me know. All right, let me. Actually, I'll put it here. So, can you see the schedule here? Before we get going, are there any questions about this organization? Okay, I'll take that as a no. All right, let me close this out. Okay, so we're going to start. I'm going to give a short lecture that covers just some background on the nucleus and chromosomes, and then Jadiel Wasson will give you an introduction to make it easier to read for the journal club, and that will include some of the methods that get used both in the papers you'll read and for some of the work that I'm going to talk about now. But right now, I just want to give you a sense of chromosomes. So if there are no questions, then I will just get this up and running. Okay, so I hope everyone can see this. And all right. So today's lecture, we're going to cover two areas. We're going to talk about how DNA is packaged into nuclei. And we're also going to talk about self-organizing organelles. And I'm curious if any of you have covered this in your courses so far. Should I assume that everyone has covered it in their courses so far? There's dead silence here.
We heard a bit about DNA packaging, but self-organization is something new.
Say what? Say it again.
We heard a little bit about the packaging of DNA nuclei, but the self-organization of organelles is new.
Okay. And how about self-organizing organelles? Is that also something new, or was that touched on in any of your classes?
As previously said, the second bullet point is new for us.
Yeah, that's new for us. Okay. Okay, well, let's jump in. And I encourage people to ask questions as we go along. Speak up if something is not clear. All right. So here is an electron micrograph. It's a slice through an embryo, and it's showing the nuclei. And for a long time, people wondered, how is DNA sitting in the cell and sitting in the nucleus? But they really had no good way of figuring it out. And so they could see that the nucleus it wasn't a homogeneous mix. It's clearly some funny structure, but what's going on? As I'm sure you learned, here is a question. Oh, I have to turn off, hold on. Let me minimize this. So, how long are all human chromosomes when lined up? That's probably something you learned. Anyone want to shout out what they think? No speakers? Just give me a number. Are we talking centimeters, meters, kilometers?
Not sure, but I think we're talking meters.
Yep, you are right. If you line up all the chromosomes, it's 2 meters. So about as tall as a person. And what about the diameter of a nucleus? All that DNA has to fit in a nucleus. And what's the diameter of a nucleus, human cell? Any thoughts?
Maybe 20 to 50 nanometers.
Say what?
20 to 50 nanometers.
It's about 10 microns. So just to give you an idea, that's about, if you imagine DNA as a fiber of silk, it's about having a piece of silk that would stretch from Basel to Aarau, and then having to package it in a tennis ball. So it's a really profound compaction. And basically, you have a length to width ratio of 1 billion. So very long and very thin. And just for you to think about, if you think about different cells in the body, what is the most impressive example of chromosome packaging in cells in a human? And a hint is that it occurs in 50% of the people watching this video, more or less. Any guesses?
Well, I guess this was the video when the chromosomes were packed. You can see them in the light microscope.
Yes, you can get a general idea. But the tightest, the smallest nucleus in the body is sperm, where you have to fit the DNA into 5 microns. Sperm is about 10-fold smaller than, say, an oocyte. And it's not enough to just, well, let's begin to talk about that. How is that done? And you learned that DNA is wrapped around histones to form a nucleosome. So I'll just recap this a little bit. In essence, 147 base pairs of DNA are wrapped around 8 histones. The H2A, H2B dimers, 2 of them, and the H3 and H4 tetramer. And so here, down here, you can see the general layout of what a nucleosome looks like, a single nucleosome, which is the nucleosome is the word for DNA plus the histones. So it looks a little bit like a hockey puck, a kind of a flattened disc. So the dimer pairs of H2A and H2B, as well as H3, H4, they use these ends of the helices, of alpha helices, to interact with the phosphate backbone of the DNA, as shown here. And so that allows them to bind many different DNA sequences. They have very little preference for sequence. And they don't have to unwind the DNA. So that configuration works well for wrapping DNA around the histones. And as you probably learned, histones have these N-terminal tails. You can see how they stick out here in this space-filling model. And these can be extensively modified. And what I'm showing here is just acetylation, but there's also phosphorylation and methylation. And these are bound by proteins. They interact with other histones to have different biological outputs. So DNA wrapped around the histones to make these nucleosomes makes this beads on a string. And so here we can see the little nucleosomes, and then this DNA in between is called linker sequences. And this gives you a compaction, it's about a third. So it's not nearly enough to package into our tennis ball, but it's a start. So histones are important for packaging and also for things like transcription, DNA repair, and so forth. But you can't just have this, what looks like a string of lights, packaged willy-nilly, or you'd end up like something like this. It would be completely unusable. So you need things to be organized in the cell, in the nucleus, in such a way that really you can use the DNA. So you need to be able to access the DNA. And this is the answer. So we're going to organize DNA into these little bun-like structures. So it looks something like this, and we're going to go through this, but just to give you an idea, this is a cartoon to show that at one level we have what are called territories. They encompass the whole chromosome. Then we have compartments. These are about up to 7 megabases in size, so very large. Then we have something called topologically associating domains, or TADs. And TADs are about 1 megabase in size. And then down here, what you may have learned about in class are loops of DNA in as illustrated here. So let's go through this. So first, territories. So the territory is for the whole chromosome. And here, this is actual data, and it's showing different chromosomes that have been marked by in situ hybridization. And you can see these are human chromosomes, so you can see them all labeled here. And what you can see is that they are placed in a discrete spot in the nucleus. So the chromosome is not spread out and it's not a rat's nest, as we say in English, but it's really this compact entity in space. Now, if you look for your pairs of chromosomes, so we're diploid, we have 2 copies of each chromosome, you can see that they're not necessarily sitting next to each other. So for example, here's chromosome 18 right here, and here's chromosome 18 over here. They are not right next door to each other, but they are compact in them on their own. People have done many of these staining experiments, and they've done them in different cells. And they can see that on average, some chromosomes tend to be in the middle of the nucleus, and some tend to be more to the exterior, closer to the lamina. And that can change in different cell types. So here's the first order of the organization, which is territories. And I should say there may be a little mixing of chromosome territories, chromosome loops, and DNA at the boundaries here, but by and large, they're self-contained. All right. The next level of organization is these compartments. And here on the left, this is a simulation of how compartments may look. So the idea is that a single chromosome can be divided into these 2 compartments, A or B. And so the red here represents the A compartment. It's a little more internal, and the B here is this blue. So what are these compartments made up of? Well, the first thing that people noticed when studying compartments is illustrated here on this diagram. So here we have a chromosome, and the blue and the red are like the blue and the red here. And you can see that it is not contiguous. So different parts along the chromosome can be red or blue, but these non-contiguous sequences cluster in space. So all the reds are here together and the blues are here together on average. People have studied then what makes up the A and the B compartment. And it looks like this. The A is the more active region of the genome. It is euchromatin. And did you guys learn about euchromatin in class? Yes? No?
Yes, we did.
Okay, good. So an A compartment is essentially the euchromatin section. It has more unique sequences, fewer repeats, active genes, but certainly not all, active modifications of histones, and it tends to be what people consider more open, although that's a very vague term. So think of this as where the bulk of the transcription and activity is going on. If we look at this part, this is generally the heterochromatin. So it has a lot of repeats. It's much more likely to be transcriptionally silent, but certainly not all. And it has many more silencing marks. It's considered more compact. It has large loops of DNA that go into making this structure. So this is the more silent part of the genome. And it makes the point that the more silent and the more active tend to be separated from each other and clustered the like with the like in space. So now we can think about this. This is the euchromatin, this clear part here, and this is the heterochromatin put here. And I should say this is an embryo, so it's still in the process of consolidating the compartments. They're still kind of mixed up, and over time they're going to coalesce more. Now, the other aspect of these regions of DNA, and you can see it a little bit here in this section, is that they go to different parts of the nucleus. And in particular, these B compartments, the orange here, tend to associate with the nuclear lamina and the nucleolus. So they form something called LADs. You don't have to remember that, but it's basically the lamina-associated sequences. And what gets them here is partly the configuration of the DNA itself. You know, it's a little bit stiffer, a little bit maybe more rod-like. But in particular, there are proteins that span from the DNA to the lamina and keep the silent regions associated with the lamina. So that gives one some organization right there. On the other hand, the A compartments tend to be more central. And as genes activate, they tend, if they were near the lamina, they will often move away from the lamina and become more internal. But it's not quite that simple. If we look up close, you remember there are nuclear pores sitting here in the nuclear membrane, and those are important for lots of things to shuttle in and out of nuclei. And these regions are associated with active DNA. So you can see it a little bit here, that this is a little bit more open. You have these dark regions of heterochromatin with the lamina shown here, and then a little bit more open here. And there's a lot of traffic of proteins and RNA coming in and out of the nuclei. And some highly expressed genes can even be tethered to the nuclear pore complex. So it's a fast way to get RNAs out of the cell and into the cytoplasm so they can be translated. Questions? Okay, I'm going to assume everything's super clear. And then here we have what are called the TADs. These are smaller. And these are a contiguous region of DNA. And they're regions that can form loops. So we can look at it here. If you imagine this is a sequence of DNA, and there are boundaries that delimit the end of the TAD. So this would be one TAD, and this would be another TAD, for example. And within the TAD, regions can loop from one to the other. And Jadiel will explain how this methodology works, and you'll be figuring some of this out yourself. But what the boundary element does is it makes sure that sequences here are less likely to interact with sequences over here, so that you get little neighborhoods that can loop and interact with one another, and that are separated from other neighborhoods over here. This is drawn as though there are 3 genes, that's the blue arrows here, but in fact TADs are often a single gene with multiple regulatory elements in the neighborhood. So how are these generated? And let's look at this here. Imagine we have a piece of DNA, They are bound by a protein called CTCF. So this is a green protein here that binds regions in the DNA and can bind on the linear DNA. So to generate a loop, we then have a ring protein called cohesin. Now, you may have learned about cohesin from its roles in replication, but this is a different role. This is during interphase. And we're looking at this ring here, and now the DNA is going to get pushed through the loop, through the ring, and we start forming a loop here. So we have CTCF and we have cohesin. And so if you keep pushing this through, eventually you hit one of these, actually two of these CTCF regions. And CTCF is a very strong binder, and so it blocks further pushing through of the DNA. And this stops this loop of DNA. So now we have a big loop of DNA, maybe a megabase in size, and this region has a tendency to interact with itself. So within that big loop, we can get these smaller kinds of loops shown here. And so here we have 3 TADs. Each of them is one of these big things. And then within it, we can get different kinds of interactions with the DNA. And so probably you learned about enhancers looping over to promoters. And so that happens within the TAD. So an enhancer region could loop over to a different promoter. And so when we think about these TADs, these boundary elements with the CTCF is very important for really setting off a particular region. Now we know these things exist in cells, the territory and the compartments and the TADs. And there's been a big discussion in the field about what do the TADs do. And people are trying to test if you disrupt the TAD, what consequences are there? And this is ongoing research. This is still happening today. And maybe some of the most interesting ideas are shown here. So this is a region in the human or mammalian genome with 4 genes. And here are the TAD boundaries. So these block interactions between the 4 genes so that you get one TAD here. And let's say this is enhancers that can interact with gene 1, but they're much less likely to interact with these other genes because these other genes are in different TADs and separated by these boundaries. And each of these genes are sitting here and involved in forming digits in the limbs. So researchers wanted to know, do these boundaries, are they really important for regulation? They knew that the genes were important for making digits. And so they knew that there were different kinds of syndromes associated when these different genes are disrupted. And so you can see some of this here. So normally, of course, we have, for example, 5 fingers on our hand, but as things got mutated and mixed up, you can get these different kinds of defects here. And this is true in humans and also has been seen in mice when the same kinds of mutations are made. And so they looked at different configurations that disrupted the boundary. For example, flipped the region. These are the 4 genes, just to give you the actual data. And so if you disrupt these boundaries, now you can start to see these different syndromes. And they found that they could recapitulate this in a mouse by disrupting these TADs. So this is some evidence that the TADs are important for gene expression. And there are other experiments where they see less of an effect. So it's been a really interesting way of studying this and getting a sense of what these things do. But our best guess is that they define neighborhoods of interacting DNA. So that is then these 4 regions, these 4 ways of folding up a chromosome so that it's still usable, but it's very compact. And these folds here and here, it's very, very compact. So are there questions on this? Anything confusing or just a question about the science? All right, then if not, then I'm going to ask you a question in a breakout. So I'm going to give you a puzzle and you think about what the answer is. All right, so here it is. You're studying this region of the chromosome. Oops. And you perform a series of experiments to disrupt the TAD domains. So in your first experiment, you mutate CTCF so that it can no longer bind DNA at the boundary. And in a second experiment, you mutate cohesin, the ring protein. So based on the looping model I just showed you, what do you think happens to transcription of this gene 2? Do you think A, that wild type has the highest transcription of gene 2, more than experiment 1, more than experiment 2, because of the enhancers here activating gene 2? Do you think experiment 1, the CTCF mutation, gives rise to higher expression, more than experiment 2, more than wild type? Or do you think experiment 2 has the highest expression, more than wild type and more than experiment 1? So think about it for a couple minutes. You can chat with other people watching this if you have a friend. And think to yourself which of these 3 options you think might be the right answer. And if you want, you can ask a question out loud. This is not so much to test as just to sort of think through how things work. So here's a hint. This was the model that I gave you. And maybe that helps in thinking about how gene 2 might be affected by these mutations and its proximity to gene 1. So any guesses, A, B, or C? Have you guys made up your mind? Do you need more time? Well, I'm not hearing anything, so I'm going to move on, but I hope you're thinking about this. So if this is the model for how it works, here's what we might imagine in the mutations. If we get rid of cohesin in experiment 2, we no longer have the ring. We're not going to form a loop. On the other hand, if we don't have CTCF, we can still form the loop in experiment 1, but we don't have these anchor proteins to tell it when to stop. So we're just going to keep pumping DNA through this to make a bigger and bigger loop. So if we think about that, then the experiment 1, where you get rid of CTCF, is likely to have the highest expression because we're going to remove this boundary. And now this enhancer element can contact gene 2 because they're all in one big domain. On the other hand, if you mutate cohesin, you have no loop at all, and the whole thing is just a linear piece of DNA. So there's no active repression of communication, but they're pretty far apart because the DNA is not in a big loop. So the prediction would be wild type, there's active inhibition of crosstalk between the enhancer and gene 2. So we'll put this as the lowest expression. Experiment 1 looks the highest because we form a loop where these regions are there together. And in experiment 2, we're not inhibiting it, but we're not bringing them into juxtaposition. So I think that would probably be in the middle spot. Does that make sense? Would you agree? Dead silence. That's a bad sign, guys. All right, well, given the amount of time, I'm going to move on to the second part and discuss organelles in nuclei. And actually, it's not just nuclei. So here, there are many structures in nuclei and even in the cytoplasm that are not bounded by membranes. So I'm guessing in class you learned about organelles that are surrounded by membranes, like the nucleus itself, or the ER, or the Golgi, but perhaps not these other kinds of organelles that do not have membranes around them. And what we'll talk about as an example is this. It's the nucleolus. So as you recall, the nucleolus is very important for making ribosomes. And the nucleolus is the site of rDNA transcription. And you build the nucleolus around this, around these sequences. Now, ribosomes are the biggest energy investment of the cell. They have a lot of protein and RNA, and there's something like 10 million copies per cell. So the cell pours in its resources to generate the ribosomes. And so it needs to do it efficiently. It needs to be able to regulate it. And so what it does is restricts it into the nucleoli. So this is a cartoon of a nucleus, and then the nucleoli sit within the nucleus. Here's an EM picture so you can see what a real nucleolus looks like. And this just gives you an idea of what happens. So within the nucleolus, you start transcribing and making the RNAs, and you actually start to assemble the ribosome itself. And then gradually it moves into the nucleoplasm and into the cytoplasm to be finally built into a functional ribosome. So to do so, the cell has to generate this organelle, the nucleolus, within the nucleus. And it's worth it doing it because really having this localized structure is very useful. So on the one hand, it can speed up reactions. Having an organelle like the nucleolus allows a reaction to go faster because you concentrate the components together. They're more likely to bump into each other, more likely to generate whatever it is you need generated. In this case, transcription of nucleoli, processing, starting to build the ribosome. They also sequester proteins in the right place and actually keep out other proteins from the organelle. So they give a level of specificity to reactions going on in cells. And of course, it allows a lot of organization so that you can make structures to physically organize the structure of cells. Now, something that people noticed with the ribosome and is illustrated here is that they are dynamic. They're not, they're different from like a mitochondrion, perhaps. They're really very dynamic. And you can see here that they can fuse together. And that happens during the cell cycle, for example. And in fact, what people have found is that the way the nucleolus behaves, it's like it's a droplet, like it's phase separated, and it behaves like a liquid droplet inside the nucleus. So you can imagine a droplet of the nucleolus here, a droplet here, and then they're merging together. So that is a very different way of thinking about organization than how we are usually taught. But it's becoming apparent that this kind of organization happens more and more. So let's talk about this a little bit. And you're used to thinking about phase separation, for example, with water, which can be in a gaseous phase, or a liquid phase, or a solid phase. It's the same molecule all the time, but just organized in a different way. And so here's an example of what one of these phase-separated organelles looks like. This is not a ribosome, I'm afraid. Sorry, not a nucleolus, but it's something called a P granule. But I just want to give you the idea of how it looks like a liquid. And maybe it's reminiscent of kind of oil and vinegar, the way they separate out if you try to mix them together. So let's think about this a little bit. And here, what we're asking is for molecules to separate into two separate states. And we're taught that this is really not what molecules want to do. They want to do this, right? This is not my bedroom. But they want to achieve a high entropy, very messy state. So what we normally think of is that if you have something organized, it's tend to go into a mixed state like this. And so we're asking, can this happen? Can something that is a mixture demix and become two separate entities? And that's what is happening in these kinds of organelles like the nucleolus. So we can think about this a little bit like this. We're going to imagine a lattice, and we're going to think about interactions between the different molecules. So we can think about the energy of, let's say, two blue molecules interacting, or two red molecules interacting, and we can compare it to this, a red and a blue molecule interacting. So this is the mixed state, and here are the separated states. And you can actually calculate it out. So you can look at the kind of energy between what we're going to call epsilon AA, which is the energy for this blue, and the BB, and the AB. So for phase separation, we need that the energy of the heterogeneous mixture is very high, and it's higher than the energy of the homogeneous mixture. So that even though this is a better entropic state, it's still more favorable to go to the lower energy state of this separation here. So in other words, the energy of the mix, if we subtract the average energy of these unmixed states, is much greater than 1. And here we're dividing it. This is Boltzmann's constant and temperature. So it's essentially, that's the unit of energy that we want to think about. And if this is much greater than 1, then it's a lower energy state to separate like this, like oil and water or oil and vinegar. Boltzmann's constant, I don't know if you've run across that before. It basically relates temperature to energy. So it's a way of converting the temperature here to energy. Now, this kind of separation here is 2 different molecules, red and blue. In fact, we can think about even more kinds of separation. And if we look at a nucleolus, we see that there's multiple. There's this blue granular component, there's this red, and then there's this green. And this is the kind of configuration the nucleolus makes. So it separates out these different structures. Here you can see what is the blue, you can see what is the green, and you can see what is the red here. And so it's the same principle that if the energy of the unmixed state is lower than the mixed, then this is what will happen, this demixing of multiple components. Any questions about this? It's probably a different way of thinking about how cells behave or molecules behave. All right, dead silence. Okay, then we're going to do another breakout. So let's imagine you have some molecule and you're looking at whether it will phase separate out of solution. So this is the normal situation. And here you have the same configuration as in experiment 1, but you raise the temperature. What do you think this will do? You raise the temperature. And here in the third experiment, you have the temperature as is in experiment 1, but you raise the concentration of your protein. So what do you predict? Do you think all 3 experiments will form separated droplets? Do you think 2 is more likely to form a droplet than 1? Do you think 3 is more likely to form a droplet than 1? Or do you think both of these guys are more likely to form a droplet compared to 1? So think about it. Think about what you would pick, A, B, C, or D. And then we'll talk about the answer. Any thoughts? What of these three is most likely to form a separated state, a droplet where the blue molecules clump together because that's the best energetically formed? Okay, well, let's walk through it. So this is what I gave you before. And here, this is a chi, it's called chi. And you can actually graph the different values for chi. And so this is this concentration that we talked about. And as you raise the concentration, you are more likely to get demixing. You can see that here. So that makes sense because you need the interactions between these molecules if you're going to form a phase separated state. So concentration matters a lot for phase separation. What about temperature? Well, if we think about temperature, it's down here in the denominator. So as you raise the temperature, this whole number is going to get smaller. And so as you raise the temperature, you are less likely to form a phase separated state. And one way to think about this is that that raising the temperature is interfering with favorable energetics of the molecule to molecule interactions. Like if you boil water and you get steam. So with more temperature, you're less likely then to be able to do this. You're more likely to do that. Oh, and this is just a cartoon to illustrate the effect of different concentrations. So based on that, we would say C. As you raise the concentration, you're more likely to get separated phase droplets of your protein out of solution. In this state, you're less likely because you've increased the temperature, your chi value goes down, your molecules are less likely to interact. Does that make sense to people? Questions? All right, let's move on then. So I've treated these little balls as just single entities, but in fact, a very important aspect of this is what we call valency. And valency describes the interaction of this one molecule with another. So here, if the valency is 1, we have one little place to interact. So these two molecules can interact here. You get a dimer. That's the end of the story. So this is considered a dimension of assembly of 0. You're not going to get phase separation if all you have is a dimer. On the other hand, let's say you have a valency of 2. Now you can begin to make a form. You can begin to get this linear string of an assembly. And that's a one-dimensional entity. And let's take another step. Once you have 3, a valency of 3, now you can really begin to make more of a scaffold, an entity. This one is 2-dimensional, right? So this has a big effect. Your valency, the places you can interact, has a big effect on whether you're going to form a phase-separated structure. In the cell, polymers are multivalent interactors. They can have many different kinds of places where they interact. So, here's a chain link to kind of illustrate this. And here we have, 5 places of interaction. So if you have a polymer like this, it can form all kinds of interactions to make then a phase-separated state. So where do we see that? Well, one place we can see it is with proteins. And let's look here. So probably in your class, if you think about the sequence of a protein, you learned that it folded into a structure such as this protein here with a very high, what should I say, degree of reproducibility. There's another kind of protein, and they're found frequently in the cell, and they're called intrinsically disordered proteins, IDPs. And what that means is that they can assume different configurations. So they don't make this one structure. They can make a lot of different kinds of structures. And as a consequence, they can interact in many different ways. So you might get something like this, where you have the same protein, two conformations, and they're interacting because they have these different valencies. And now as you increase that, you can start to get a big kind of interaction scaffold in 3 dimensions. Now, protein does this, but RNA can do it as well. And what people are finding is that RNA is often the nucleator of a phase-separated state. So you can have a strand of RNA that then binds to proteins and nucleates the onset of this phase separation. And the proteins are then these disordered proteins or have domains that are disordered and that can then interact with RNA, but interact with each other to build this kind of 3-dimensional structure. And so here is just 2 examples where this one, for example, has 2 different proteins and they form a droplet. And here we have one that's an RNA and a particular RNA binding protein, and it too can form a droplet. And it does so by binding the RNA. And you can see that has a valency of, let's say, 3 or 4, and also interacting with each other here in this disordered portion of the protein. So this can begin to nucleate one of these phase-separated states. So DNA, this is protein and RNA, but people are thinking now that DNA can do this as well. And that DNA or chromatin, so this is our beads on a string again, that here by interacting with different kinds of proteins, it may form something like a phase-separated state. And there's a very big debate on what the nature of this would be. Is it truly phase separation or something like it? And so this depends on the kinds of proteins that interact with the chromatin fiber and whether they begin to nucleate this kind of a structure. And so in the nucleolus, the thinking is that's exactly what's going on. Okay. So I'm going to turn off the this, and that's my introduction to the nucleus, to this phase separation that seems to happen a lot in the nucleus. And we have a few minutes left for questions, and you can type it into the chat because now I can see the chat, or you can speak up, and I'll just wait here. I can, I'm happy to talk to people one-on-one, but I'm also happy to talk to the whole group for any questions you might have on nuclear organization, phase separation, and so forth. No questions. Well, do people want to turn on their video for a moment so we can see the class? All right. I'm glad you're still there because it's very quiet.
Maybe you should have a look on our pictures. There were a lot of thumbs up during the lecture. I don't know if you haven't recognized them.
I'm sorry, what?
We can do something like a thumbs up with our emoji. And when you ask a question like, is everything clear? We can do those thumb ups. And then I don't know if you have seen that.
Well, I mean, I don't want to really step through the whole lecture again. But let's see. Because I can't, the problem is, let's see. If I put myself into presenter mode, I cannot actually see your thumbs up. I can hear you, but I cannot, I can't see you. So that's kind of a problem. What I can do, but it's not ideal. Hang on. But if you're very brave, you can just ask me a question. So let's see. All right, I'm gonna do it this way. Now the problem is I can really only see a few at a time. I can't see the whole class. All right. So if you have a question, either say stop or I don't know. So we went through this, this. Oh, no, we didn't go through that. This. Nucleosome. Putting together a nucleosome. Nucleosome histones interacting with DNA. Oh, I wonder if I can see the, hang on. Oh, can't do that. Tails, beads on a string. What we're trying to avoid and how the cell fixes things. I should point out that you have a PDF of this whole lecture on Adam. So here's the overview of chromosome folding. Territories. Compartments. Compartments are non-contiguous, even though they're together in space. They correspond to euchromatin, heterochromatin. So now we can begin to assign identities to this picture. B compartments sit at the lamina. A compartments are inside. But active DNA does associate with pores. And then we talked about TADs. And TADs are these neighborhoods that can interact with each other. They can form loops, for example, enhancers to genes. But they tend not to cross the boundaries to the next TAD. then this is how you make a TAD into a loop domain. You have this CTCF boundary factor and cohesion ring that can go around chromatin. And so you can pass this through and begin to make a big megabase loop. And then eventually you get this structure. Now this can interact. This is the TAD, and this is the TAD. But it's separated from these other TADs. And so this is just to illustrate again that we have the CTCF boundary that kind of blocks the interaction in space. Here again is the example of a region in the human genome of these genes that are very important for digits in limb development, and where it's important that the enhancer not talk to these other genes, and that's with these TADs. And then here is the region of, region where if we start to change the organization, we have the same sequences, but we're moving these TAD boundaries around, we start to get these malformed digits. And then we did a breakout. What happens is we start to disrupt CTCF or cohesion. So we're disrupting either the green protein or the yellow protein. What would that do to this region here? And so the prediction is no cohesion, we don't get loops at all. And no CTCF, we get loops, but it passes through, keeps making bigger and bigger loops. So its answer is B. And then we spent time looking at these organelles and structures that do not have membranes surrounding them. So we focused on the nucleolus, which is the site of where ribosomes are made, as shown here. And I'm going to skip ahead. We looked at this movie of what one of these organelles can look like, where it really looks like a liquid. And then we discussed going from a mixed state to a lower entropy phase separated state, and what are the parameters to do this. And so we started looking at the energy of the interactions between similar molecules and different molecules, and we can actually calculate out the conditions chi. Yeah.
I have a question concerning the TADs.
Okay.
Is it absolute? So there should be no interaction between two TADs, or is it dependent on the gene expression, so on the required genes?
It's not, so let me go back to the TADs. It's not an absolute. People will see occasional interactions, but it's lower than if you remove this. So there's a tendency to keep the interactions here and much lower likelihood to interact outside the TAD.
But is it sometimes required?
Say what?
Is it sometimes required to have those interactions, or is it really not necessary and also always malfunction?
I know of no example where it's required. I don't know of any example where it's required that a region interact outside of the TAD. I think it's considered more of something that maybe happens, a low frequency, but it's not really what's
okay.
What's should be going on, I guess. Okay, so to go back here, we calculated out to say that when chi is much greater than 1, that is to say the energy of this interaction is not favorable. compared to the like interactions, that then we will get this separated state. And that can be more than 2 molecules. And in fact, in the nucleolus, we have several of these states. And we did another breakout, asking what kinds of things influence phase separation. And what we find is higher concentration, more interactions, that favors demixing. And also higher temperature disrupts these interactions, and so it favors mixing. And then we looked at the valency. I'll just jump ahead. The more places you can interact, the more kinds of assemblies you can make. And where we see this a lot is in polymers. So they can form structures like this. And in particular, we see that then with proteins that have these intrinsically disordered structures shown here. But we can also get it with RNA. So RNA plus these disordered proteins can begin to nucleate these phase separated states here, or just protein, and ultimately chromatin. So DNA is also a polymer. It binds and interacts with many different kinds of proteins. And depending on how those proteins interact, you can begin to set up a distinct state, not bounded by a membrane, but it is an isolated kind of entity. Okay, so that's the whole lecture speeding by. And I'm going to again stop the share so that I can see the chat. And if you have questions, either speak up or send me a chat. And why don't we take a 3-minute break, and then we'll start on Jadiel giving you an introduction to the papers and telling you a little bit about methods also. Okay? Okay.